Hello everyone, so today we're doing cellular respiration part 3. So we talked about these already, and now we're going to move into the metabolic pathways of oxidation. So now we're going to be looking at the aerobic pathways, glucose oxidation from ATP, and the metabolic pathways. So before we looked at the function of glucose, and we also looked at fermentation and anaerobic pathways, so now we'll be focusing on those aerobic pathways. So as we mentioned several times, these metabolic pathways harvest energy of carbohydrates, so glucose. So today we'll be looking at aerobic conditions. So again, there's complete oxidation. The waste products are H2O and CO2, and we can have a net of 32 ATP. So just keep that in mind and try and count along as we go through. So what are the aerobic pathways? So first, we have pyruvate oxidation. So when ovate 2 is present, then we enter the citric acid cycle, and then we have the electron transport chain, or ETC. So again, citric acid cycle is also known as Krebs cycle, or tricarboxylic acid cycle, or TCA. Just different names for the same thing. So in cellular respiration, this permits complete oxidation of glucose to energy trapped in ATP in the presence of O2. So as we remember, we had partial oxidation when we did glycolysis and fermentation. But now, with oxygen present, we can have complete oxidation. So again, we have this graph here where we have glycolysis, where pyruvate, and we either have O2 or do not have O2. So again, this went to fermentation, so we're not looking at today. So now we have O2, and now we're going to look at what occurs in the mitochondria. We're going to be looking at acetyl-CoA and the citric acid cycle that occur in the mitochondrial matrix. But yeah. So first, before we get to those electron transport chains and acid cycle, we need pyruvate oxidation. So pyruvate oxidation is a link between glycolysis and the citric acid cycle, or CAC. Pyruvate oxidation only occurs in the aerobic pathway, and it occurs in the inner mitochondrial membrane in eukaryotes and occurs in the inner plasma membrane in prokaryotes. So here we can see on the plasma membrane, pyruvate oxidation, and in humans, we have in the matrix, or the inner mitochondrial membrane. So pyruvate oxidation is a multi-step reaction catalyzed by an enzyme complex called pyruvate dehydrogenase complex, which is also called PDH, pyruvate dehydrogenase. This reaction occurs in the mitochondrial matrix in eukaryotes, which we'll focus on. So the reactants of pyruvate oxidation is pyruvate from glycolysis. So again, this occurs times 2 because we produce 2 pyruvates. We need an NAD+, and we need coenzyme A, which is also referred to as CoA, which you can see how this is a bridge to the citric acid cycle, where we actually end up with acetyl-CoA. So we'll see that this comes in. CoA will come up quite a bit, especially when we're going to biochemistry as well. So in pyruvate oxidation, we have the important complex, pyruvate dehydrogenase complex, catalyzes 3 reactions. So the first reaction is pyruvate is oxidized to acetate and converted to acetyl group, and this releases 1 carbon dioxide, CO2, and energy. Second, energy is captured when NAD+ is reduced to NADH again in this scenario. And 3, the remaining energy is captured when the acetyl group, so remember acetyl, combines with the coenzyme A, yielding acetyl-CoA. So again, here's a reaction. We have our pyruvate. We have here we go through. Energy is reduced. NAD+ is reduced to NADH. We have the release of that carbon dioxide. Coenzyme Q is added, binding with this. So then we have the formation of acetyl-CoA. Here's a CoA group. So again, we can see inner membrane space in the matrix. So here we can kind of see pyruvate goes down, loses that carbon, becomes acetyl-CoA in a much simpler view. So again, it's a multi-step reaction catalyzed by an enzyme complex, PDH. This enzyme complex is attached to the inner mitochondrial membrane. It occurs in the mitochondrial matrix in eukaryotes. Again, the reactants are that pyruvate, 3 carbon, NAD, which gets reduced to NADH, you see here, and coenzyme A. So our final products are acetyl-CoA, which is a 2-carbon molecule, because remember, We lost that carbon from pyruvate. There'll be 2 molecules per glucose molecule. So remember, this will all be times 2. And then we have NADH in hydrogen. Again, 2 molecules per glucose molecule. Then we also have energy, which is stored as NADH in acetyl-CoA. So the next step after pyruvate oxidation is the citric acid cycle. Like I said, the Krebs cycle or tricarboxylic acid cycle. This completes the oxidation of glucose to CO2. So this occurs only in aerobic pathway, and occurs in the mitochondrial matrix again, and occurs in the cytoplasm and prokaryotes. So examples we'll be looking at are in the humans. So in the citric acid cycle, we use the energy in acetyl-CoA, which again is 2 carbons. So keep in mind the carbons, as this is important in the citric acid cycle. This drives the reaction of acetate, which is a 2-carbon molecule, with oxaloacetate, a 4-carbon molecule, to produce citrate, 6 carbons. This is the first reaction of the citric acid cycle. So this is essentially where oxaloacetate and acetate and acetyl-CoA work together to form the citrate, getting that name citric acid cycle. So this is followed by a series of reactions. So you've probably seen the citric acid cycle. So it's a big circle with different areas, a bunch of steps coming off. But anyways, this is followed by a series of 8 chemical reactions, which citrate, the 6-carbon molecule, is oxidized to oxaloacetate. So essentially, this citrate is used to produce that oxaloacetate again, and then that's used to create another citrate. So again, it goes in that circle. Citric acid cycle. So it's a continuous cycle. So this oxaloacetate 4-carbon is regenerated with the release of 2 molecules of CO2, 2 times released at twice, 1 time. So again, if you go to the carbons, we're going from 6 back to 4. This completely oxidizes acetyl-CoA into 2 carbon dioxide molecules. So at the end, this produces 2 acetyl-CoA per glucose, which are used in glycolysis, producing 4 CO2s at the end. So again, the reactants are acetyl-CoA, which is 2 carbon from pyruvate oxidation. So again, this is times 2 per glucose. We have NAD+, or FAD, which is flavin, which we'll talk about some later and more in biochemistry. We have our ADP and inorganic phosphate to form energy, and as well as water, or H2O. So here's that big figure that I was talking about. So again, we have our acetyl-CoA, which is 2 carbon from the pyruvate oxidation. So here we have our oxaloacetate merge with our acetyl-CoA, which is 4 carbon and 2 carbon, forming that 6 carbon citrate. So this citrate goes to isocitrate, and in this reaction, NAD is reduced to NADH, and we also have a CO2 lost. So there's 2 areas where we lose CO2 1 time, so CO2 and CO2. Then we go to alpha-ketoglutarate. Again, another NADH is produced, and we lose the CO2. Then we go to succinyl-CoA. So in this succinyl-CoA, we can see the coenzyme A comes back. Then we have, in this reaction from succinyl-CoA to succinate, we have the formation of ADP, of GTP and ATP. Then from succinate to fumarate, we have that FATH being formed, which that's that flavin we're talking about here with the FAD. Then we talked about we need that 1 water molecule. So we need the fumarate to malate, we need H2O to get added, and then again another reduction back to our oxaloacetate, and then the cycle repeats when another acetyl-CoA comes in. So again, the products are carbon dioxide times 2, released twice separately per acetyl-CoA. So again, this would be times 2. So total would be 4 CO2s. We have NADH, which are 6 molecules per glucose. So again, 3 per cycle, but 6 because the cycle is happening twice at the same time by each pyruvate. Then we have 2 molecules of FADH per glucose molecule. We also have energy, which is stored in NADH and FADH. So as we'll talk about, these NADH and FADH go to the electron transport chain, because as you can see, these are carrying energy. And then we have 2 ATP, and we have 2 ATP are generated per glucose molecule from the citric acid cycle. So here's essentially the energetics. So here we have our change in free energy. So we talked about glycolysis, where you have this buildup, then the drop. And as we can see, we keep getting more down and down and down. So essentially, these become very energetically favorable. So these are in the cytoplasm and the mitochondria. So here we have the citric acid cycle. We have the citrate forming. These are kind of the steps of the citric acid. So remember, we talked about there's 8 steps. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 8, sorry. Then we have the formation of oxaloacetate, and then again, this kind of goes back up, and we do that again. So now, moving to the final stage, the electron transport chain. So again, it only occurs in aerobic pathways. It's also in the mitochondrial membrane, in eukaryotes, and in prokaryotes, again, it's in the plasma membrane, as you can see here. So electron transfer chain is essentially the recycling process involving the electron transfer, and this involves oxidizes reduced energy carriers NADH and FADH. So these become oxidized back to their NAD and FAD, and in so they're transferring electrons, and several proteins move these electrons to special carriers that can use these electron potentials to create ADP. But we'll get to that soon. So through the electron transfer chain, we have the recycling process involving electron transfer, hence its name. We oxidize the reduced energy carriers, NADH and FADH. So the reactants we need are those NADH, like we said, from glycolysis and pyruvate oxidation in the citric acid cycle, FADH from the citric acid cycle, and ADP in phosphate, and O2. So again, we need O2 because it's aerobic metabolism. So the energetics. So each NADH generates 2.5 ADP, and each FADH generates 1.5 ADP. So for every NADH and FADH that enters the electron transfer chain, we get 2.5 for NADH and 1.5 for ADP. So we'll see later that, like we said, we produce a certain amount of NADH, and then we'll go in and net this. So this involves 4 large proteins. So here are these complex 1, complex 2, complex 2, complex 3, and complex 4. These are those large proteins, and here we have a bunch of different enzymes working. So these are small proteins, or the cytochrome C, which you can see is CYT, and we also have non-proteins with our ubiquitin, which is the Q. So the free energy level decreases through the electron transfer chain. As you can see here, we slowly decrease the free energy, and this free energy is stored in ATP, and these excess electrons are accepted by O2, forming water. So as I mentioned, O2 is the final electron acceptor of the electron transfer chain. So after all this electron transport, O2 is where all the electrons end up, then we form water. So the electron transfer chain involves 3 parts. First is the respiratory chain, which is the electron transport chain, or ETC. So in the respiratory chain, this releases energy as electrons move or fall from high to low energy levels. So as we saw, these electrons are falling from energy level to energy level, and then finally very low to oxygen. This energy the energy released as it moves from these energy levels is used to do work, and it involves moving protons. So a lot of these things will move protons up and out. We'll talk about that later. So second, we have the active transport of protons. So protons are moved out of the matrix, which creates a concentration/charge gradient across the membrane. So that's very important. This charge gradient creates a source of potential energy that can be used to make ATP. Then finally, we have facilitated diffusion of protons. So these facilitated diffusions of protons are through the proton channel back into the matrix, releasing energy that is captured as ATP synthesis. This is secondary active transport. So we use this first active transport to push them out, and then we pull them back in, and we use that secondary active transport to produce ATP. So here's a summary of the whole complex. So you can see it gets quite complicated if you want to really study in detail. But we have our complex 1. So when we talk about those hydrogen protons being moved in and out, here's some things called like coenzyme Q, coenzyme QH2. Here we see our NAD comes in, they're oxidized. So NADH enters that complex 1, getting that 2.5 ATP, I believe. And FADH enters that complex 2, yielding that 1.5. So it's one less step for FADH, but less ATP. Then we go to complex 3. Here we can see a key component, cytochrome C, which transfers electrons from complex 3 to complex 4. Here we see the electrons move. Here we have our oxygen, the final electron acceptor, producing our water. When this happens, we have because of that proton gradient, and then these protons move through. They enter through the ATP synthase, which helps create ATP by merging these ADPs and inorganic phosphates, forming ATP. And again, ATP is pretty much used by every cell for energy, so we have this happening at crazy amounts of levels at a time to constantly produce energy for our body. So as I said, we have a proton gradient, causes the protons to diffuse back into the mitochondrial matrix through the membrane channel protein. So here's our intermembrane space in the mitochondrial matrix. Sorry. And then we see this goes through the membrane channel protein, the ATP synthase. So again, we build them up, and then they all come through, and then we use them to build ATP. This is facilitate diffusion of protons, provides energy for ATP production. Essentially, the summary here is oxidative phosphorylation. So essentially, we're using this oxygen to create this huge gradient, and then we're using it to phosphorylize ADP into ATP. So through the electron transfer chain, we have the recycling process, which involves electron transfer. Again, there's so much detail we can get into here, but we'll get into more detail in our biochemistry. But just keep in mind, if you're studying like the MCAT or something like that, you would definitely need more detail than this. Again, we have the oxidation reactions, which reduce the energy carriers NADH and FADH. Again, our reactants, you see here, reactants from glycolysis, pyruvate oxidation, and citric acid cycle. Then our products are NAD plus and FAD, which go back up into the cycles. Then we have our H2O and our ATP used for energy. Then again, energy, our net ATP produced is a total of 28. The reason it's not that 32 we talked about before, because we had to invest some, so we have a net gain of that. But it depends, so we'll talk. So again, overview. The products of reactions become reactants of the next reaction. So here we can see the linked chains. So again, we have 30 ATP produced. We can see here this FADH is produced, used here. This ATP is produced, can be used here, is produced here. This NADH is used here, produced here. Same thing with here, used here. So all these things are connected. They're not just produced for no reason. So even coenzyme A, we know coenzyme A is produced to form that acetyl-CoA, and then that goes back in to form that succinyl-CoA, then it goes back into the cycle, and again and again. So yeah, it's a very interesting pathway. And it's honestly quite fascinating. You can spend hours studying it in detail, which you might have to. If you take any more advanced courses at university, you'll definitely get into more detail. And I definitely recommend it because it's just, it's essentially the fundamentals. Even if you go into quite a lot of detail, like you might not have to remember every enzyme or, aldolase does this. You just need to know like the basics, like so you already know what's happening. And I think the key way to do that is through knowing the reactants, the pathways, and the products. So if you can follow the products more than anything, and you know what goes in and what comes out, just by that you can kind of know what happens. But yeah, anyways, I definitely take a look at more detail if you would like. So how are these metabolic pathways interconnected? So the end products of partial oxidation, which we talked about, which are glycolysis and fermentation of glucose through anaerobic fermentation, still contain unused energy, so only a small amount of ATP is produced. Net is 2 ATP. The end products of complete oxidation of glucose through aerobic cellular respiration converts energy to ATP, leaving very little unused energy, with a net of 32 ATP. Essentially, here's a summary of the entire reactants. So we have our glucose, and 6 oxygen go in, 6 carbon dioxides come out, and 6 H2O and so I can just zoom in there for you guys. So again, glucose goes in, 6 O2s. So this is from we get this oxygen from breathing. These 6 carbon dioxides, which are then removed as waste. We have our 6 H2Os, which are probably just recycled and used in different reactions. And then our 32 ATP for energy. So again, our electron transfer chain gives us 28, which I mentioned, plus the 2 from citric acid cycle and the 2 from glycolysis. So netting 32. So again, that's it, guys. So that's our 3 parts of cellular respiration. As I mentioned, really interesting topic. Recommend you study in detail, especially if you're looking for something like the MCAT or for any higher level courses. But yeah, hope you guys learned something. Take care.